Hello, everyone. I would like to welcome you to this week's edition of Extracellular Vesicle Club. EVClub was established by Ken, but has now grown into an event of the International Society for Extracellular Vesicles. My name is Metka Lenassi, and I will be your host for today. And now it is my great pleasure to welcome today's speaker, Ina Nikonorova from Maureen Barr Group at Rutgers University. Maureen's group has a strong background in C. elegans research, which they use to model human genetic diseases and also study fundamental questions in biology, with recent focus also shifting on EVs. And I'm excited to hear more about that. I encourage everyone to place any comments or questions in the chat box during the talk, and it will be possible to unmute yourselves at the end and ask some of those questions also in person. And now over to you, Ina.
So thank you so much for inviting us to this series of wonderful EV clubs. Today I'm going to talk about how we use C. elegans to understand fundamental biology of a human. I'm going to start with this slide that shows three types of EV biogenesis mechanisms. And today I'm going to focus my talk on specifically on ciliary EV biogenesis. And the cilium is a special structure that present on almost all cells of the human body. It is a cellular antenna that transmits signals. It has a microtubules inside and a special gating mechanism at the base of the cilium. So this gating mechanism prevents simple diffusion from the cell into the cilium, and the lipid composition of the cilium is also very different from the cell membrane. So thus EVs that released from the cilia are fundamentally different from all other EVs that are released by the cells. And just to talk more about, a little bit more about cilium, just to introduce how important it is. Dysfunction of the cilia cause ciliopathies, diseases that affect many organs. And some of the examples are depicted here. So it can be a complete inversion of left and right axis. Your heart will be positioned not on the left but on the right side of the body. Can be infertility associated with the immotile spermatozoa, non-motile spermatozoa, or severe brain abnormalities, retinal degeneration where rod and cone cilia are affected, complete loss of smell, cysts in liver and kidney. It can also cause obesity and skeletal malformations like a very narrow rib cage or more than 5 fingers polydactyly. We think mechanisms for all the ciliopathies just not always understood well. And we think that ciliary EV biogenesis might be a component of that contributes to ciliopathies. When cilia is non-functional, the ciliary EV signaling might also be dysfunctional and maybe contributing to manifestations of the ciliopathies. And so cilia EV interactions can be observed in electron microscope. And here you can see transmission electron microscopy and scanning electron microscopy of cells from bile ducts. This is actually pathological conditions. Too many EVs are observed. And this, you can see a process of EV budding from the cilium of cultured cells, ciliated cultured cells. Or you can also see this spermatozoa, maturing spermatozoa in the reproductive tract of a turtle. So those round things are actually cilia, flagella of the sperm cells, and some of them contain many vesicles inside the cilium. Because these images are static, it's impossible to say whether those EVs are actually going from the cilia or they just coming to the cilium and fusing to the ciliary membrane. So that's why it's beneficial to use some model organisms where this EV biogenesis can be directly observed in the microscope. And I'm going to introduce our model, C. elegans. On this image, you can see male C. elegans. It's a round worm. It's about size of a comma, 1 millimeter long. And I'm going to talk specifically about ciliated cells that we study. So somewhere here in the brain of the worm, there is a special neuron that sends dendrite to the very tip of the male nose. And at the end of this dendrite, it's called sensory neuron. At the end of this dendrite, there's a cilium. Similar mini brain is located in the tail of the worm. It also sends dendrite into special ray structures. You can see there are 9 of them in the tail. At the end of this dendrite, there's also cilium. And so this neuron that I just described, they actually responsible for mating behavior, initiating mating behavior of the male. And thus they're very crucial, their function is very crucial for reproduction. And the important protein that's expressed in this neuron is actually called PKD2, which stands for polycystic kidney disease 2. And this is our Ortholog of Human Polycystin 2 protein. And so if we look closer with super resolution microscopy at the very tip of the cilium in those ray structures, you can see that PKD2 comes in the environment in the form of EVs. So those ciliary vesicles. And in this picture, you can see ray 7 and ray 5 are in this process of release. And so that's how we can directly observe the whole process in the microscope. And just to make it relevant to human health. So in human, dysfunction of this PKD2 protein would eventually lead to severe form of polycystic kidney disease, and it also cause left-right abnormalities of the human body. Whereas, so basically it's a dysfunction of primary cilia, whereas in C. elegans, it leads to dysfunction of the sensory cilium, and such worms with non-functional PKD2, they're not able to initiate mating behavior. And we actually use this mating behavior as a readout of functional state of the cilium, sensory cilium, so it's not lethal as opposed to mouse models. So in human, this polycystin 2 interacts with polycystin 1, and in a worm, likewise, PKD2 interacts with LAV1, which is Ortholog of Human PC1. So those proteins that act in the complex. So there is a evidence for physical interaction of this PC2 and PC1 in human, and we have evidence for genetic interaction of PKD2 with LAV1. So in human, it was shown that urinary vesicles contain PC1 and PC2 on the same EVs. Here you can see small beads are actually PC1, and smaller, large beads are PC2, and smaller beads are PC1. And we do similar analysis in our lab, where LAV1 antibody would stain small vesicles in purified from the culture of C. elegans. And we also do colocalization studies. We do it a little different. We study it with fluorescent reporters. And currently our lab fully transitioned to CRISPR-Cas9 generated tags. So that means the fluorescent tag is introduced through genome editing, so they reflect endogenous levels of these proteins. And here on this picture, you can see tail of the worm. And at the end of each ray, in this case, 1, 3, 4, 5, 7, 8, 9, you can see the cilium. I'm going to zoom out. Ciliary base, you can see both PKD2 and LAV1 colocalization. And they also colocalize in the cilium, and they also go in the same EVs. You can see that the EVs are released from these rays and this ray into environment. So this actually preliminary data, and they come soon in bioarchives. The whole method of CRISPR editing transformed our lab and our ability to analyze ciliary biogenesis. This was introduced by Jonathan Walsh. So because we have such an incredible model, we can analyze genetic pathways of this conserved polycystin, analyze this conserved polycystin pathway, right, using genetic methods available in C. elegans. And so the goal of my project was first, because we know that PC1 and PC2 on the same EVs, because they conserve, they probably have other conserved components that we don't know about. And so the goal was to identify basically the proteome of ciliary EVs. So when I joined the lab, we had to collect that many ciliary EVs from the worm culture. And worms eat bacteria. So they co-cultured with E. coli on agar dishes. So I used this strain, PKD2 labeled with GFP, so it's only expressed in the males, the direct male mating behavior. So those amazing partners, hermaphrodites, do not express PKU2-GFP. They don't have those neurons. So I cultured a lot of worms. We collected EVs through serial centrifugations on accution. And then we used gradients, either loaded from top or from the bottom of the tube. And we calibrated them overnight in those gradients. And then I looked at each fraction to see which fraction would contain green vesicles or PKU2 labeled vesicles. And normally enriched fraction would look like that, so that those vesicles were clearly visible. And the typical profile from this isolation would look like that. So this fraction number on x-axis and y-axis would have actual number of PKU2 EVs that I was able to see in the field of view. And the right axis would be the amount of protein that is in each fraction. And so you can see the profile. So the PKU2 EVs come in two peaks. Actually, we call them light and heavy, slightly different density. That's very reproducible. And the majority of protein from our preparations were coming at larger densities. And as it turned out later, it's mostly bacterial outer membrane vesicles, probably. And because we have EV audience today, I'm going to talk a little bit about technical details of this method. So if that's of interest to someone. So, but first we confirm the presence of the vesicles in our light PKU2 enriched fraction and heavy PKU fractions. And those are some cute small vesicles that are not visible at this magnification. So they all contain EVs. So you can see their own structures and they all have a protein associated with them. So our top loading actually was better because bottom loading approach where the EVs has to float from the bottom of the tube to upward didn't resolve two peaks well. And this fraction, it has a abrupt density. So basically contain both light and heavy EVs. And so we tried to troubleshoot it a couple times and were not able to. So we proceeded with top loading and we did two more biological replicates. And this time we decided not to combine technical replicates. Those are technical replicates are at sort of y-axis. So those were run basically at whatever run at week 1 and week 2 would be biological replicates, and whatever travels in different tubes would be technical replicates. So we didn't combine technical replicates and submitted fractions for proteomics separately. So each fraction submitted is labeled with an error. So you can clearly see two peaks in every fraction, more or less. But before submitting to proteomics, we wanted to know how the protein profiles look like on gel. And so here's the typical profile. You can see that this majority of the protein, which we think is OMP, outer membrane protein, highly enriched here. It's about 37 kilodalton. And it also leaks sort of in other fractions, especially the heavy PKD2 enriched fraction. And so when we submitted it for proteomics, we actually choose to cut out this band out of the gel to potentially increase our discovery rate of the proteins that are not OMP. And so this is all the fractions submitted, and the band that was cut out is marked with yellow brackets. And so the concern of our reviewer was that we get rid of all the 30-40 kilodalton proteins, and we analyzed like post factum when we already determined the protein. So we didn't see any decrease in 30-40 kilodalton proteins in light fraction as compared to heavy fraction. And actually this profile approximately resembles the whole protein profile of C. elegans. So our technology to increase the discovery rate, it doesn't lead to any loss of a protein. It's just potentially allows us to discover more proteins. So the analysis came back was actually 2,888 proteins, which is about 13% of C. elegans proteome, and that's a huge number of proteins. And we realized that they not only come from ciliated neurons, but from other tissues. And literature says that yes, other tissues actually do shed EVs. The cargo for those not always known, but they can be observed in the microscope. And they were reported for reproductive tract intestine, hypodermal cells, embryos, and probably glands and excretory system also able to release EVs, we don't know. But we were focused on ciliated neurons. So I'm going to talk about those, how we figured out which cargoes out of those 3,000 probably come from ciliated neurons. So for that, we had to do a little bit of mining. So we collaborated with bioinformaticians and developed this app that is specific to our data set and that allows to see which cargo is likely coming from which tissue. And I'm going to talk a little bit how this my EVOme app works, what approach it uses. So basically in C. elegans, aside from those tissues that I already told you about, the ciliated neurons, there are different types of ciliated neurons. They have different transcriptional signatures. And just to introduce C. elegans to you, I have this is the head of the worm. And the neurons that are exposed to environment are labeled here on this diagram. So first, it's IL-2 stands for inner labial 2. On this wonderful headshot of the worm, you can see them in yellow. So each bud has a ciliary tip that protrudes into environment and able to release ciliary EVs. So the blue amphid, so-called amphid neurons, they are chemosensory neurons, and they located their number of neurons. They're located in those pockets, and the opening of that pocket are indicated in blue color here. And the green neurons, they cannot be seen here because this is hermaphrodite, and we discuss male, so CEM neurons, cephalic male neurons present only in the male worm. You can see I put male tail here. R and Bs, they also express polycystin as cephalic male. So you can see there's cilia over here, also protrudes to environment. And you saw EVs, I showed earlier how exactly they release EVs. So those are star labeled very tip of the cilia, right? So because these neurons has different transcriptional signatures, and there is a data set of single cell transcriptomics of C. elegans. So basically, the transcriptional signature of all the cells of C. elegans is identified. And so we combined our data set of proteins with single cell transcriptomic data sets, where for each protein, we saw its expression in TPM values, transcript per million, in every cell of the worm. And so we started scoring our cargoes based on this expression. So basically on x-axis, I'm going to show you this IL-2 neurons, TPM value divided by all neurons. Basically, that's enrichment, whatever expressed in IL-2 neurons, over all neurons taken together. And y-axis would be ciliated neurons, broadly speaking, all of them, as compared to all other tissues. And so all our cargoes were spread on this plot, and we can see that there are three different groups, mostly enriched in IL-2s, mostly enriched in sort of other ciliated neurons that are not IL-2s, and the ones that expressed in both. And so I only labeled cargoes that were already known to be released by those neurons. So we knew we are on the right track with this approach. And we tried to validate some novel cargoes that were on this plot. And so I'm going to talk about these 4 cargoes that turn out to be ciliary EV cargo. So that's MCM3, which is Ortholog of Human MCM3 protein. ENPP1, it was not known gene. We had to assign name for it, and we named it after human NPP1. So this tetraspanin 6, TSP6, Ortholog of Human Ct9 that you're all familiar with, and SID2 that I'm going to talk about a little bit later, that it doesn't have an obvious human ortholog. So our validation protocol included creating a reporter on native promoter, injecting to the worm, and imaging with high resolution. I'm going to show you some images of those cargoes. And first is this SID2 that is unique to nematode. Nematodes, so SID stands for actually systemic RNA interference defective. And this is actually a double-stranded RNA transporter that expressed in worm intestine. So if worms eat double-stranded RNA, that will be bound by SID2 and internalized into intestine, and actually leads to genomic silencing of complementary sequences in the whole body. So this process is actually conserved in human. It's just C2, the very step of this process. We didn't find any orthologs of C2. And the Nobel Prize was awarded for discovering this double-stranded RNA interference mechanism in 2006. That was discovered in worm and widely used right now in all the systems. So what we did, we used genome editing to add tag to this C2 protein. And the power of C. elegans is that right now we can, first of all, track endogenous protein, endogenous gene expression by using this reporter. And also C. elegans has a robust functional assays where we can assess whether our reporter is functional at all. And so this is just example how we did that. So when they absorb double-stranded RNA, so the gray color shows the neutral double-stranded RNA, and yellow is something that is highly lethal. So knockdown of MEX3 gene is lethal, and so you can see that our reporter actually was able to absorb this double-stranded RNA, and no progeny was produced. This is total progeny of 3 hermaphrodites, 3 mothers that were eating this kind of RNA. And if you compare this to null mutant, you can see that double-stranded RNA that was lethal was not actually absorbed and it didn't initiate any silencing. So both of them produced a normal amount of progeny. And so we discovered that C2 not only expressed in intestine as was reported previously, also specifically expressed in ciliated neurons that we were interested in. So that's an unbiased discovery. So you can see here male tail and all its cilia labeled. So I'm gonna 1, 2, 3, 4, 5, and in each cilium you see PKD2 GFP or reporter, a reference reporter. It's very enriched at the very tip of the cilium. I'm gonna zoom a little bit at the very tip of the cilium. And C2 in M scarlet labeled with M scarlet and magenta, it's located in the base of the cilium. In ray 5, it's a typical setup, but in this ray, you can see that occasionally C2 travels from the base through the ciliary shaft and into the environment in the form of EVs. So this is actually first report we were very excited about, where double-stranded RNA transporter for some reason is exported from the neurons, sensory neurons, into the environment. So we're just working on the functional significance of that right now, trying to figure out why would you need that. So same process happens in the head of the worm. This is cilium, ciliary base with a lot of PKD2 at the base and ciliary shaft, and this is the head of the worm. So in the environment, you can see stream of these PKD2 and GFP. PKD2 and C2 and scar. So C2 is almost gone from the base here. It's just zooming, so you can see better those vesicles. So the second cargo that we're excited about was also nucleic acid binding protein, which is mini chromosome maintenance 3 protein. It's a DNA binding protein, and it acts as helicase normally during replication. So it forms hexameric structures with MCM1 through 6, but archaeal MCM filaments, MCM orthologs actually form filaments, not hexamers, that bind the single-stranded DNA, unwind DNA. So we were looking for connection to the cilium, and we actually found that in human MCM proteins actually do show signs of ciliopathies. And for example, MCM2 associated with autosomal dominant deafness, that would indicate that cilium in the inner ear is not functional. And in zebrafish, MCM2 knockdown also would manifest ciliopathy phenotypes. And so we created the reporter. And as predicted, MCM3 was highly abundant in dividing cells, and it locates at nuclei, so that's consistent with replication function. Also, the only cells that expressed MCM3 and when they were not dividing were ciliated neurons that express PKD2. So on this image, you can see ciliary base with MCM3 highly enriched in the base, and it also goes through the shaft and in the environment in the form of vesicles. So whether function of MCM protein in the cilia somehow associated with its DNA binding ability, we don't know. It's a subject of further investigation, but no doubt that MCM proteins are important for ciliary function across, in evolutionary conserved manner from human to worm. So the next cargo that we validated was Ortholog of Human Ct9 that is common in EV preparations from human fluids, right? So that's a tetraspenin. Tetraspenions have a special crystal structure, a special structure which is inverse cone, and they promote EV biogenesis. When they concentrate on the membrane, they start bending the membrane. And so we also looked for connections of CD9 to cilia function, and we found two reports where first evidence that CD9 was cloned first time from bovine ocular ciliary epithelium, where rod and cone cilia would be located. And the second function also sort of speaks to its importance specifically in the retina. So if you injure retina, they never recover in CD9 knockout mice, but they're able to recover in wild type mice. And actually in wild type mice during injury, CD9 is expressed at high levels, so which speaks to its role in ciliary function. And also while we were working on this project, tetraspenin 6 was independently discovered by other group as a ciliary V cargo in C. elegans, and it participates in neuron-glia interaction possibly in C. elegans. So imaging of this, it turns out that tetraspenin 6, as predicted by our bioinformatic mining approach, it's expressed in different types of neurons, not the ones that express PKD2. So I'm going to show you how those cilia are organized. It's basically a bunch of different cilia in one pocket over here. It's called amphids. And they all have opening into environment. This is ciliary pore. And there is also other cilia here that is highly branched. So you don't see many branches on this cartoon. It's actually highly branched. You can see all those threads like structures. Those are branches of this AWA cilia. This is a chemosensation cilia. So basically, but the point is here, so this is ciliary pores. And from those ciliary pores, you can see a lot of tetraspenin 6 EVs coming into the environment, which validates our proteomics data set and our mining approach to determine which cells produce which cargo. And so the last cargo I'm going to talk about today is very interesting. So it's actually called ENPP1, which is this long name. Basically what it does, it's a phosphodiesterase that would cleave triphosphate nucleotides over here and release pyrophosphate. So ENPP1 in human causes excessive, if it's mutated, it causes excessive ossification due to dysfunctional metabolism of these phosphate groups. But what it's also able to do, it's actually upregulated in viral infections, where it's able to cleave this exotic nucleic acid sequences, 2' 5' connected, and some cyclic nucleotides, such as C-GAM that is formed in viral infection. And so the human NPP1 is highly homologous to the worm NPP1. And in worm, we saw it being in the cilium. So here is the sensory cilia. NPP1 is a scar. You can see large bubbles coming from the ciliary membrane into the environment. So I want to point out the NPP1 is very abundant in human fluids, and it's abundant in EV preps from human fluids, but it was never associated with the cilia function. And so our report is indicative that it might also be play a role in cilia of the human at some organs, at least. And another finding that I'm going to talk about, so I'm going to finish with the cilia story, right? But once we made this ENPP1 reporter, aside from this sensory cilia, we also discovered that it's expressed in another tissue, which is male reproductive tract. And here you can see the mature sperm, mature spermatids would be stored over here in the body. And then upon ejaculation, those have to travel through vas deferens and into the vulva of the mating partner. So this vas deferens has large vacuole-like structures, and on their presumably membrane, we saw a lot of NPP1 and sort of a dust inside the vesicles. We speculated that those might be actually exosomes, and those large vacuoles might be large multivesicular bodies, but the reviewers said you cannot say that without electron microscopy, so we had to remove the statement. But this is the video that shows you Z-stack through these vacuoles, and so those dust speckles, they kind of moving freely inside those vacuoles, which we found very fascinating to see. And so the question was, once they express in vas deferens, are they actually transferred to the male partner during mating? Will they be like carriers of information or some kind of support to spermatids during copulation? So we conducted experiment where we crossed fluorescent male with non-fluorescent hermaphrodite. This is, but just to understand the results of this experiment, just want to introduce anatomy of mating partners like hermaphrodites. They modified females and they have uterus that has many, many eggs, 13 in this case. They have ova that would move, in this case, from right to left and go through spermatheca where self-sperm is formed and into the uterus. So they can produce progeny by themselves without males. However, if there are males and this hermaphrodite is inseminated, the sperm will travel from the vulva area into the spermatheca and would fertilize the ova that would come through the spermatheca into uterus. So what we did, we took non-fluorescent hermaphrodite and our NPP1 labeled male, we crossed them, and we actually discovered that whatever stored in those vacuoles is actually expelled with sperm into the uterus and stays in the uterus, presumably in the form of EVs. But we also didn't do microscopy, electron microscopy, so we cannot say for sure that those are vesicles. But I mean, all the facts says that those probably exosomes. And with this, so this experiment shows how we use this C. elegans model to determine target tissues of vesicles by just tracking fluorescence and using microscopy. And because those pathways are concerned, we think we can use this model to learn a lot of things about human EVs and their target tissues and mechanisms of targeting. So with this, I would like to end my presentation. And as a conclusion, just want to say a huge thank you to Maureen Barr and the whole lab. And we worked as a team, and it's a very exciting project. And we're actually expanding it right now, validating more cargos. So basically, I want to say this is a large data set. We provide methodology how to predict cell-specific EV cargo out of a complex mixture of candidates. That can be applied to C. elegans to other tissues, or maybe in the future we can use similar methodology to do similar analysis in human mice. And we also, the main discovery of the paper was that nucleic acid binding proteins are actually cargo of ciliary vesicles, which I think has a potential for regulatory function of cilia affecting other tissues.
So thank you, Ina, for sharing with us this very interesting study. And I see at the moment there's no questions in the chat box. There is one, and I'm just going to do just a second so that I can unmute. Okay, so I allowed all the participants to unmute themselves, and I will just let Clotilde Thierry ask the first question. So please go ahead.
Hi, thanks very much for this very nice, beautiful images, beautiful presentation. So I had a few questions, actually. I don't know if I can ask them all, but one of them was first, when you show these pictures of these big vacuoles that you think the vesicles that originate from them are exosomes. It's not so clear to me, because when the picture looked like they have they have external vesicles, but not internal vesicles, these vacuoles. And so, yeah, what you're showing here, these small vesicles that are outside the big vacuoles. So why, I mean, and I don't understand where these vacuoles would fuse and release their content to make exosomes. So maybe you can just clarify that. And then I had a quick question about the other potential tetraspanines that you would have identified by proteomics, if you had identified other tetraspanines.
So the first question about the structure is, I think that this one area is a membrane bound, a lot of NPP at the border, and those super small things, they're tiny vesicles, exosomes. These guys, so I'm not sure what they are. I think they might potentially fuse with this later in the development, but I don't have evidence for that. And so we look at this from lateral view. So there's other set of similar vesicles on the right side. I mean, this is the left and not. So there is similar row on the right. So there are two rows, like they form a channel. They call cubical cells. So presumably when ejaculation happens, you know, somehow the signal ends up in the uterus. So you can imagine that maybe those MVBs fuse and release all the exosomes into the channel.
But
we don't know. Yeah.
But you don't have the ENPP1 in the internal structures of these vacuoles.
Yeah, no, they filled with this. It's a lot there. It's just, it's a lot brighter on the membrane, and you think it's kind of dim inside, but it's very bright inside. It's just, you know, matter of brightness and the amount on the membrane. And,
okay.
And sometimes I see like a row of vesicles budding. I mean, you can see, I mean, it's a static process. I was not able to find, to see dynamics, picture dynamics. It's like a grape structure budding from membrane inside.
Okay.
Like series of connected vesicles, like a grape. And the second question about tetraspanines.
So yeah, no, I was just asking about the tetraspanine because as you are probably, I don't know if there are other tetraspanines in C. elegans, but that the fact that CD9 is in human, I mean in mammals is mostly at the plasma membrane, so it somehow fits with the fact that your EVs bud off the tip of the cilia and don't come from MVBs. I was wondering whether there is an equivalent of CD63 in C. elegans, or maybe I was also thinking of tetraspanin 4, which is found as very enriched in these other types of vesicle called migrasomes that form also in sort of tips of maybe not cilia, but other structures. So I was wondering whether you had any
Tetraspanin 4 would be human name, right?
Yeah.
So overall, C. elegans has more than 10 tetraspanins. That's called TSP something. So out of those, we have just 4 tetraspanins in our preps. I was not able to validate others. So I just didn't make reporters for them. But we have another interesting protein that expressed specifically in PKD2 insulated neurons. It does look like tetraspanin, but it's very unusual tetraspanin. So normally you would see transmembrane 1, 2, 3, 4 would be short loop and long loop. So that tetraspanin is inverted, so it would be long loop exactly like that here, and the short is here. So we're currently working with that one, but I don't have an answer yet. So that would be predicted to be specifically co-expressed with PKD2, and we think it might be important for ciliary EV biogenesis.
Nice, thank you.
Okay, thank you. And Esther has another long question, so I will just let her ask it live in person.
Thank you. Yeah, Ina, thank you very much for this interesting presentation. And when looking at these very beautiful pictures that you showed, something occurred to me, and I'm not really sure what I'm seeing. So I thought I'd better ask you. Because when you look at the PKD2 EVs, they seem to be all lining up like kind of beads on a string. Whereas when you look at the other stains, they seem to be randomly localized around the tip of the cilia. So I was wondering, do you know anything about whether they also seem to be on different subsets, although you have not shown all the co-localizations, but so how do these come out of the cilia, and why do we see these different patterns, you think?
Yeah, thank you for this question. So we recently discovered that this release of vesicles is stimulated by mechanical pressure from the cover slip, probably. So interesting structure of the male tail. You can see this ray 1. You can see very tip of the cilium. It's because it's facing cover slip on this particular image. And ray 2 would be, and it's dorsal, so it opens on the dorsal side. Ray 2 is ventral, it opens on the other side of the fan, and that's why we don't see vesicles coming from there, because cover slip is not touching that. So ray number 3 is lateral, so it doesn't touch the cover slip fully, only probably. So, and you see the spread of vesicles random here. And so ray 5 is dorsal, ray 4, I think also. I'm not sure about ray 4, but so we think that you see a straight line because when you put cover slip on the worm, it might slide a little bit, and that might provide a sort of a tangent on the membrane, cellular membrane, facilitate EV release, and they will be like sticking to the cover slip a little bit and not spreading too much on the slide. Okay.
But I did not see these lineups for the other markers.
Yeah, so other markers, we're not sure they colocalize with PKD. I think they not colocalize with PKD2. We don't see this colocalization similar to like LOV1, right? So those are different. So they're not on PKD2 vesicles. There's like something else, and right now we're trying to figure out what the mechanism for those would be. Yeah, so those will be diffused. And
Ina, yeah, can I also see it clearly, that the pink is not in the lineup that you see for the green?
What, Maureen?
Hi, Esther. I'm just going to interject here. So Jan Wang had a paper showing that cilia shed at 2 different sites, the tip and the base. So there could be 2 different biogenesis machineries. And then I'm going to hand Ina, could you comment on your turbo EV proteome using PKD2 and what you think is going on with EV subtypes?
Yeah, so we recently, because none of the cargoes, validated cargoes, had perfect colocalization spike PKD2, we decided to enrich further in a way. So we attached biotin ligase to PKD2 to do proximity labeling and collected those vesicles. And so right now, like it came with roughly 10 proteins. And right now we're starting validation of those. And like I did first 5 reporters and they do colocalize perfectly with PKD2. So right now we have a better method of figuring out different EV subtypes, right? Because the density centrifugation will still give you a crude mixture of different things. But if out of those you just, purify only those that proximity labeled by biotin ligase is specifically targeted to one subtype, we will be able to figure out, the groups of cargoes that go on one type of disease. Am I doing it right?
Great. Yeah.
Wait.
Thanks for the answer. Yeah, I think that's a very interesting approach to take. Yeah.
So maybe just a follow-up question to that. I'm not so familiar with C. elegans EVs, but I was surprised that in, let's say, mammalian cells, you would see these vesicles formed throughout the membrane. The membrane would kind of like be almost blabbing, or at different sides there would be release of EVs. But here it seems like there's this hole or something that just pushes the vesicles out. I mean, is a mechanism known about how these EVs are formed or where they are formed? Because obviously they just are transported through the cilia to the release point. It's just like the whole thing seems a lot different to what I'm used to. So maybe if you have a comment, if it's even known.
Yeah, so what we see under the microscope, EV coming from one single pore. And if you look here, so the yellow bud is covered with cuticle, and only small opening at the one. So that's probably why you see directed. When we do serial section and analyze under electron microscopy, we do see EVs sometimes surrounding the cilium, like in that pocket. So whether they shed from the side of the cilia into that lumen is the question. But like under a microscope, you would not see that. You would only be able to see whatever is coming from the pore. Am I understanding your question, right?
Yeah, great. So I guess that the other parts are have some kind of layer that is not permeable to EVs, so they are protected. So
yeah,
that's
so
why
covered
EVs are
with
just
cuticle,
okay.
right?
Okay, great. Thanks. Yeah, a lot.
I mean, we do
understand it
see
a lot
sometimes. So for this CM neurons, the at the base, it's so like lumen is so extended that you can see actual budding of the vesicles from the membrane at the base of like below the cilium itself. So those vesicles are also in lumen. Whether they release, if they would be, if it's possible, they release in the environment ever. So they will also be coming through that pore.
Okay, thank you.
But some of them are too big. Yeah, some smaller ones.
So just a small question. What are the sizes of these EVs? Do you see like the whole range of sizes, or are these just more smaller EVs?
Yeah, I didn't include the diagram, but the sizes range from 50 to 200 nanometers. Clearly something we see with NPP1, they blow like a larger bubbles.
Okay,
yeah,
so
I mean,
majority
of them are within 50 to 200.
Thank you. And now back to you, and sorry for for interrupt. How many of the nucleic acid binding proteins are membrane associated? So is this probably she's asking, is this a general, is this something like a general, generally, are they generally present on the, are they generally associated to membranes, or is this something that is unusual?
I think something that like SID2 would be one of a kind sort of a protein. I think it's unusual. We did find like over 200 plus acid binding proteins. A lot of them didn't display any signs of membrane tethering. Those were argonauts, Y box binding proteins, ribosomal proteins, transcription factors, tRNA synthetases. Yeah, I mean, this like membrane bound is sort of a unique SID2. I don't know any other one from the top of my head.
Okay, thank you. And are there any other questions?
Yeah, this is Phil asking me.
Okay.
I have a question. This is really fascinating stuff. This is a creature from another world. And we're going to learn a lot. I was wondering if the male vesicles bind to the female or are taken up by the female.
So when we made PKD2-GFP male with the hermaphrodite unlabeled, we do find deposition of those PKD2 EVs around the vulva area. Yeah, so we think they might, I mean, our speculation is they might protect vulva from like antimicrobial agent in a way. But we tried to test this hypothesis right now.
That's
yeah, so the beauty is, yeah, we can see fluorescence.
Very interesting.
So I have another question. Hopefully, and others can still, of course, go ahead and ask their questions. But you said that you are producing. I mean, you were growing these worms on a plate with bacteria growing. Like bacteria was probably the food. Do you know if bacterial EVs or bacteria communicates with the C. elegans, and do you think that it affects the release of EVs that you're studying? I mean, how is this communication? Do you think that this is something that is going on?
Yeah, that's an interesting question. Yeah, I do think they communicate. I don't know how. I think vesicles might be part of this. We found tons of OMP in our preps, despite this excision and enrichment and everything else. Yeah, so actually we deposited raw data. Maybe someone at some point will try to figure out what bacterial vesicles doing to C. elegans. I don't know. Yeah, it's interesting.
Yeah.
It's hard to test though, you know?
Yeah, it's just like it's you know, interspecies communication. So it will be interesting to see how this affects those studies.
Yeah, although I have to say, yeah, bacteria, the total number of proteins in C. elegans about 20,000 as in humans. In bacteria, it's roughly 5,000 that encoded in the genome. In our preps, we found roughly 1,000 bacterial protein, which is like 20% of bacterial proteome. I mean, we didn't analyze the data in our paper, but the raw data is available.
For enthusiasts of OMVs, right? Okay, so any quick question from anybody? Otherwise, I would really like to thank you again, Ina, for taking your time and explaining about your work today here. And of course, also to everyone who attended and joined this meeting and also, of course, asked these very interesting questions. And I'm sure that we'll hear more from you and from your future studies. So good luck with everything. And of course, have a nice afternoon, evening, wherever you are. So bye-bye.